Wonderful. Thank you very much. It's a pleasure to join you this morning, and thank you very much for the invitation. As you kindly introduced me, I'm a geneticist based in Manchester and have an interest in rare inherited conditions, and would like to tell you a little bit about the work that we've been doing on Perrault syndrome over the last 10 years. So this is the first family that I saw with this rare condition 10 years ago. 3 young sisters, all were referred to me with severe hearing loss and with varying degrees of ovarian insufficiency. So you will see that the first young woman had had 2 children, but she had a premature menopause at the age of 23. Her sister had started her menses at the age of 14, but by the age of 18, these had stopped. And the youngest sister, on imaging, had absence of the ovaries. And so the 3 sisters had varying degrees of ovarian insufficiency, but all of them had severe sensory neural bilateral hearing loss. All 3 sisters had some very mild degrees of intellectual disability. They had small skull sizes, and they had some white matter changes on their brain scan with evidence of cerebellar atrophy. So this appeared to be ovarian insufficiency, hearing loss, and some cognitive neurological features. We recognized that this was consistent with this rare condition, Perrault syndrome, which had been first described 70 years ago. It was first described as a condition that looked very similar to Turner syndrome, but these women had a normal carrier type with hearing loss. This was then followed up a number of years later with a description of a number of young women with this condition. The hearing loss in this condition, in the cases described, can be very variable to affecting the high frequency hearing loss, all hearing ranges, or more severe at certain ranges. The ovarian insufficiency, as we've seen in our very first family that I described to you, could vary from premature menopause before the age of 40 to complete ovarian dysgenesis. It's unclear if males are affected with fertility problems, and there's still most males that present with this condition will have hearing loss, but most males appear to be fertile. We know that there are very few cases of this condition that have been described, but we think that there is an under-ascertainment because most individuals with hearing loss won't be asked about their ovarian function, or if their hearing loss occurs at a very young age, it will be before they've reached puberty. As I said, the neurological features are often progressive. They can occur in up to 40 to 50% of people and can affect a number of different conditions. So can lead to ataxia, intellectual disability, and peripheral neuropathy. So 10 years ago, a group in America, led by Mary Claire King, first published a paper describing 2 sisters with variants in a gene called HARS2 that was consistent with this diagnosis of Perrault syndrome. She subsequently went on to describe 2 further families who had Perrault syndrome, but also neuropathy and ataxia, but they had changes in a different gene, a peroxisomal gene called HSD17B4, which is also known to cause this very severe condition called bifunctional protein deficiency. So even though this is a very rare condition, already 2 different genes have been described in 3 of the original families. When we went back and looked at these 2 genes in our 3 sisters, we didn't find any variant in either of the published genes. And so we thought that there may be a 3rd Perrault syndrome gene. We undertook a process called autozygosity mapping, where we used the information by the fact that the sisters were related, the parents of the sisters were related to each other, and we're looking for shared regions of genetic information. And we found a single region which was found to be shared by the 3 affected sisters, but not in an unaffected member of the family. We then looked at the sequence data using exome sequencing in that region and identified a variant in a gene called clpp. This missense variant, changing an amino acid, was absent in healthy controls. It was present in the 3 sisters homozygously, but not in the unaffected family members, and both parents were carriers or heterozygous. We worked with Tom Friedman in the United States, and we identified 2 further families, one of 4 sisters, one of 2 sisters and a brother, who also had changes, one a missense change and one a splicing change in this same gene. And we showed that the splicing change led to a change in the transcript. We showed that this protein is present in the ovary, and also expressed significantly in the inner hair cells of the ear. Since our original description, there have been a number of different gene changes throughout the gene, both missense changes and also more severe variants in this gene associated with Perrault syndrome. Men with Perrault syndrome with changes in clpp appear to have a zoospermia, which is different to the relationship and fertility in males with changes in the other genes that have been associated with this condition. Subsequent to our discovery, a group in the Netherlands showed that people with more severe changes in this gene can have severe white matter changes in the brain with epilepsy and developmental delay, showing that there's a variable clinical presentation. What does clpp do? Well, we know that it's a very important protein in removing unfolded proteins from the mitochondria. Subsequent to our work, a group in the Netherlands identified 3 unrelated Dutch females in unrelated families where the parents weren't known to be related to each other. They found a variant in a gene called errol1, another missense change. So these women had variable degrees of sensory neural hearing loss and variable degrees of ovarian insufficiency. Interestingly, like clpp, errol1 is another mitochondrial gene. Subsequently, another gene was described to be associated with Perrault syndrome. 2 families with variants in a gene called lars2, which, like HARS2, is also a gene that's important in mitochondrial function. Variants in lars2, more severe variants, have also been associated with a very severe childhood disorder characterized by hydrops, anemia, renal disease, cardiac dysfunction. So a pattern is emerging of mild variants in these genes causing Perrault syndrome, more severe variants associated with severe childhood problems. Another gene was then described by Mary Claire King's group called TWINK, which is a gene that we know can cause a condition called mitochondrial DNA depletion syndrome. Here, children have very severe spinocerebellar ataxia and progressive external ophthalmoplegia, but milder variants in this gene are associated with hearing loss and ovarian insufficiency. So, what did we know so far? I've described 6 genes which have all been found to have changes in which can cause Perrault syndrome. which is really surprising considering Perrault syndrome is thought to be such a rare condition that so many different genes can all lead to the same condition. We know that all of these genes are really very important right through evolution as they're conserved to yeast. And we know that if you knock out these genes, that is incompatible with life. So Perrault syndrome and the other conditions I've described to you are only found in individuals with hypomorphic variants that reduce the function of the gene or change the function of the gene, but not with complete absence. As I said, we know that all of those genes, except for HARS2, have been associated with a very mild feature, which is Perrault syndrome, the hearing loss and ovarian function, to much more severe metabolic and neurological problems. So these 6 genes that we describe, 5 of them all fit into what we call the mitochondrial translation pathway, the production of proteins within the mitochondria at different stages of that pathway. So here's TWINK, HARS2 and LARS2, errol1, and clpp at this last stage of the processing. HSD17B4 fits into a different pathway called peroxisomal beta oxidation. Our recent work has shown this and another gene that we think is relevant to Perrault syndrome and how they can be linked into the mitochondrial pathway, likely through a process of prenylation. So we were working with a number of people to describe changes in these 6 genes. So people from around the world would contact us if they felt that they had a patient with Perrault syndrome, and we would undertake genetic testing and finding changes in these known genes. But one family stood out. This is 3 young women from Israel, from a Palestinian family that were consanguineous. The 3 women all had absent ovaries, hypogonadotrophic hypogonadism, very mild intellectual disability, and as you will see, very severe hearing loss. We undertook that same autozygosity mapping process where we looked for shared regions in the affected sisters that weren't present in the unaffected members of the family. And by doing that and then combining that with sequencing, we found a single genetic variant which had never been seen before in healthy controls and landed in a gene which we knew was very important for mitochondrial function, called PRORP. So PRORP is the gene or the enzyme that is made by it that cuts the RNA that is made by the mitochondria, so that you'll know that the mitochondria has its own genome that is transcribed in a single circular way, and to then cut that RNA, it's cut at 2 places, at the 5' end and the 3' end, to make the individual RNAs that encode the mitochondrial proteins or mitochondrial RNAs. And the 5' end is cut by this process, by this complex called RNase P, which is made up of 3 proteins and the RNA. It was known that variants in these other 2 genes led to severe health problems, but no variants had ever been found in PRORP. So variants in TRMT10C led to a severe health problem with lactic acidosis and deafness. Changes in part 2 of the complex in SDR5C1 led to a severe infantile neurodegenerative condition. But as I said, PRORP, or as it's also called MRPP3, had not been found to be associated with any health problems. After our original description of a family, we worked with other colleagues in Canada, in London, and in Spain to describe 3 further families. One little boy just with hearing loss. He's only 10 years old, so we don't know about his fertility. In this young boy, who's only 1 year old, he had severe changes on his brain and hearing loss. And then, interesting, in a consanguineous family from Spain, we saw that they had a change, a homozygous change in the gene, which had affected the mother. She'd inherited change from her, both her parents, but her husband was a carrier for the change, and so therefore 2 of their children also had the condition. So even though this is a recessive condition, we can see 2 generations being affected in a pseudo-dominant pattern. And they had severe white matter changes, but as you can see, the mother was fertile and had no ovarian problems, showing that there can be variability with changes in this gene. Not every woman who has a change in this gene will go on to have infertility. When we looked at the different variants in this gene, we found that they all lay in the same region, predicted to affect the enzyme activity of the gene, and therefore the ability of the protein to cut the mitochondrial RNA. We showed that in the 2 patients here, F1 and F2, that there were lower levels of PRORP compared to healthy controls. And we also showed that there was a reduction in the patients in complex 1 and in complex 4 of the mitochondrial respiratory chain, consistent with changes in the mitochondrial genome. We undertook northern blot analysis, which you can see in the healthy controls shows normal transcript splicing. But if you look at the patient, here you see a number of different products of different sizes, showing that there's incomplete cutting of the mitochondrial RNA. We also showed in an in vitro assay how this tRNA processing is reduced. So this is the normal wild type, and then for each of the variants, we showed lower levels of enzyme activity, reducing the amount of transcript splicing to variable degrees for each of these missense variants. And it looked like the patients with the most severe phenotype had the most significant reduction in this tRNA processing. We then published this work just last month in the American Journal of Human Genetics, so adding to the numbers of genes associated with Perrault syndrome. We still have a number of unsolved cases, and last year another gene called GGPS1 was also associated with ovarian insufficiency, hearing loss, and in some of these patients there was also muscular dystrophy. This gene, as I mentioned before, GGPS1, is involved in this process called protein prenylation, a post-translational modification to the protein, which potentially links it in to how proteins are made in the mitochondria, and links it in to variants in this gene, SD17B4, and also patients with mild variants in the peroxisomal genes PEX1 and PEX6, which can result in hearing loss and potentially ovarian insufficiency. We saw another patient, this is a young woman from Portugal, who presented with hearing loss and ovarian insufficiency. And she didn't have a change in any of the known Perrault syndrome genes, but in a different mitochondrial gene called RMND1. And we know that this is a gene that had previously been associated with kidney problems. When we looked at our patient, she didn't have any kidney problems at all, but this was a clearly known pathogenic change in this particular gene, expanding further the number of genes associated with Perrault syndrome. As you are all aware, hearing loss and ovarian insufficiency are both relatively common health problems. And so sometimes you may have a situation, as in this case, where a young woman presented with hearing loss and ovarian insufficiency, but subsequent genetic investigation found a change in a gene called claudin 14, which was homozygous in a known pathogenic variant, which explained her hearing loss. She had homozygous changes in a different gene called SGO2, which we believe accounted for her ovarian insufficiency. So it was that combination of changes in two different genes that led to this blended phenotype, rather than her having a diagnosis of Perrault syndrome. So what we know is that 8 genes are described accounting for Perrault syndrome. They still only account for 50% of reported cases. In our laboratory at the moment, we've discovered 5 other genes that we believe are responsible for Perrault syndrome. Of those, 3 other genes are known to disrupt mitochondrial function, and the other 2 weren't known previously to cause any changes in the mitochondria, but we've shown that they're expressed in the mitochondria, showing that studies of human illness can show new roles for proteins and genes that were previously characterized, but new biological insights can be provided. Do all these genes fit into a single pathway? Well, we don't actually know about that, and what we're very keen to do is to try and undertake work that allows us to think about a single treatment process rather than having to target each of those genes by gene therapy or some form of replacement. As you will all be aware, there are many syndromal disorders where changes in genes are associated with ovarian insufficiency. We know, however, that many genes that are important in mitochondrial function, not just the ones that I've described with Perrault syndrome, but other genes can, like CARS and NARS2, can lead to ovarian insufficiency. So wherever you see a patient who's got ovarian insufficiency and other health problems, be they affecting their hearing, their vision, their muscles, their learning ability, we should think about syndromal causes of ovarian insufficiency. There will certainly be more genes encoding the over 1,000 different proteins that are important in the mitochondria that will result in ovarian insufficiency. And one of the next things that we should be thinking about are, can changes within these mitochondrial genes affect women and be a cause of non-syndromal ovarian insufficiency, so where there is no other evidence of any other health problems? The big question for us is, why does mitochondrial dysfunction result in ovarian insufficiency? We don't fully understand that. In fact, we're only scratching the surface of that. People touch on the fact that many mitochondria are found in the ova, and that they are the largest cells in the body, and they're very mitochondrial rich. But in many ways, that on its own doesn't explain why this dysfunction should be so specific to ovarian tissue. And I think in the future, we will see lots of opportunities for treatment as we continue to understand the genes that lead to this rare condition. I'd like to thank all my colleagues who I've worked with in Manchester over the last 10 years studying this rare condition, particularly my postdoc Lee Domain and my colleague Professor Ray O'Keefe, Professor Rob Taylor, who's a mitochondrial expert, and Professor Tom Friedman, who's a hearing expert in America. Thank you to all my funders for supporting this work, and thank you so much for listening to me this morning. I'm happy to answer any questions.
Thank you. Thank you, Professor Newman, so much. always difficult to treat patient with ovarian insufficiency. And I think it's better to know what is the reason of ovarian insufficiency. And some of these patients may have Perrault syndrome, but it's a rare disease, as you say. Do you think that this kind of patient can be treated with plasma-rich thrombocyte or stem cells injected directly in the ovary?
So that's an interesting question. I don't think that anybody has tried that. My sense is that one of the approaches that looks most attractive is something called chaperone treatment. And that is because most of the genetic changes that we have found so far are missense changes and result in protein instability. And these chaperones can maintain the stability of these proteins. And if we just raise the function and activity of these proteins just slightly, I think that will have a significant positive effect. As you said, it is a rare condition, but in a few minutes I will be going to my clinic and I'm seeing a patient this morning who has this condition. So they do exist, but we need to be very open and thinking about recognizing these patients. And if you and colleagues have patients in your clinics, then we would be very happy to work with you to try to find the specific cause of their diagnosis.
Thank you. Thank you very much. Can we have other questions from the floor?
Hello.
Hi.
Hi. It was very interesting. And I also think about, do you think it's considered to search for this condition in people which are, we know that they have hearing problems, like we here have a school for that condition and many other things. Do you think this is can help these women to,
Yes, no, I think that's a really good question. So we know that for some of the women, they have variable ovarian insufficiency. So if the diagnosis is made at a young age, if they have hearing loss, then you do genetic testing and show they have Perrault syndrome. Number one, you can give them hormone replacement therapy to help them move through puberty. And number two, you can potentially, if there is some ovarian reserve, you can harvest their eggs and then store them until they're at an age where they may want to make decisions about pregnancy. So I think there's a real value in undertaking testing for these genes as part of a panel in women and in men with hearing loss.
Thank you.
Any other question from online auditory also? Professor Andreeva.
Thank you very much. Very interesting lecture. My question concerns these mitochondrial dysfunctions. It's very interesting in some cases of Perrault syndrome, but I understand that you have a lot of knowledge about rheumatoid arthritis. So my question in these cases with premature ovarian insufficiency rheumatoid arthritis, do you think that this mitochondrial dysfunction could be one of the reasons of this?
So that's a very interesting question. I don't think there's compelling evidence that mitochondria are significantly at the cause of a rheumatoid arthritis, but clearly there is a downstream effect on mitochondrial function in patients with rheumatoid arthritis. So I think it's an effect of the disease that you get mitochondrial dysfunction rather than the cause. Whether that is related to the ovarian insufficiency that you see in some individuals with severe early onset ovarian insufficiency, I think that is less likely. I think there are probably other mechanisms that are more relevant.
Thank you.
I would like to ask our participants of online Professor Yonakova have a question, I know. Did you hear me, Maria? Are you from Manchester United? Professor Newman.
No, that's a very good question. I've lived in Manchester for 30 years, but I support Liverpool. And so this is a big stress for me. I don't like Manchester United at all.
It's not good because our scientific program director is from Man United. What about Professor Yonakova?
Yes,
do we have connection? Okay, the floor is yours.
Yes, sorry for the delay. Professor Newman, thank you very much for the lecture. It was really interesting. It is a challenge for us, the patients with premature ovarian insufficiency, as we are reproductive specialists. And my question is, can you give us more practical recommendation in patients with unexpected premature insufficiency and reproductive plans? Which are the most common genetic diseases and testing that you recommend to perform before to start to try to help this type of patients? And the second part of the question is, if we detect some genetic disease as they are related to some severe problems, could be related with severe problems in the offspring of these patients, what kind of consultation to give them regarding the pre-implantation genetic testing of the embryos of these patients, or testing of the pregnancy if they conceive spontaneously, in order to reduce the risk of severe disease in their offspring? Thank you very much.
Thank you very much. Excellent question. So in the UK, for a woman with premature ovarian failure or insufficiency, we would routinely undertake fragile X testing and a karyotype. That is the baseline. It's really important, though, as I said, to check if they have any other health problems that may give you a clue that this may be a syndromal diagnosis rather than isolated ovarian insufficiency. Increasingly, I would undertake an exome analysis, and then I would screen the exome for a panel of genes that I know are associated with ovarian insufficiency. But that is not routinely done. That is just my practice. For young women with ovarian insufficiency who do become pregnant, I do not believe that there is any significantly associated increased risk to their offspring, particularly for these situations where there are recessive conditions like Perrault syndrome, unless the father is a carrier for that condition. So the child will be an obligate carrier if the mother is affected, but they will not be affected themselves unless the father is a carrier. So most situations you can be very reassuring that any type of prenatal intervention is not required.
Thank you, Professor Newman, for your time. And I will invite you in our afternoon session where the genetics question will be discussed with the genetic specialist, not with reproductive specialist. Thank you again. It's a pleasure to hear your topic.
Thank you so much. Bye-bye.
Bye-bye.